Am I all set up? Everybody see my slides okay?
You are good to go.
Great, thank you. As Kevin indicated, I'm Joel Ellis. I'm the Director of Toxicology at Recursion. First off, I would like to thank the NTSAD for inviting Recursion to this wonderful event. So similar to Ruben, this is kind of a coming out party for Recursion. This is the first time that we presented a lot of this information in a public forum like this to patients. So again, thank you for allowing us to attend and participate. What I would really like to do today is talk about our approach to drug development at Recursion and how it's enabled us to work on rare diseases in ways that that really, as an industry, historically have not been possible. At Recursion, we actually currently have 4 active clinical programs as a result of our approach to drug development. And among those is our GM2 program. So really what I would like to talk to you today about is our unique approach and how this approach to discovering drugs at Recursion and how it enables us to work in rare diseases. As many of you know, rare diseases are incredibly challenging from a drug development perspective, particularly rare diseases where there really are no medical therapies and the ecosystem is such, and as evidenced by the previous speakers, you know, there really is not a pathway that's well paved, you know, to get these drugs to the patients and approved. And this is particularly true in cases where the diseases have subgroups in patient populations who are diagnosed at different ages, have different rates of decline, and have slightly different medical histories. Just a few things that I'm going to probably bring up throughout this discussion. Just a few words, you know, definitions. I will use the word drug candidate, and that simply refers to, right, the drugs that we're evaluating. They're not available on the market, and they're, you know, they're still in process as far as being evaluated for drug development. I will also refer to in the talk machine learning or artificial intelligence. I will use them interchangeably. I am by no means a technology expert, but really what this boils down to is we're using a supercomputer to take massive amounts of data, and that allows us to make predictions. And then obviously for just drug development, I really break this up into 2 segments. We have our discovery or our early drug development, and then we have our later stage actual drug development phase. So there's 2 phases that I'll refer to in this talk. So who or what is Recursion and what do we do? We're a relatively small company, small team, about 200 to 250. We're still growing. But with the small team, we do an incredible amount of research and collect an incredible amount of data. We run more than 1.5 million experiments every week. And we do this because we have streamlined our systems and our procedures. We use robotics to move and handle a lot of those experiments. So we're very efficient in the way that we run our experiments. And because we run so many experiments, we obviously generate a significant amount of data. Currently, and this is rising obviously every week, we've generated 7.7 petabytes of data. And just for reference, and it's a reference that was really meaningful to me, you know, imagine taking all of the high definition movies ever made in existence, and imagine how much data that is. And that's roughly about the amount of data that we're currently at as far as what we've generated to date. And based off of all this data and these experiments, we've developed disease models greater than 500, covering all kinds of indications from genetic to cytokine and you know, different indications and modalities. And as I mentioned, resulting from these models and in our discovery system, we actually have 4 active clinical stage programs. And then in addition to that, we have an active pipeline of greater than 30 programs that are in our discovery phase of development. So how do we do this? Well, we have a pretty extensive tool belt that we use. All of this is internally generated and specific and purpose built. Biological tools, chemistry tools, cell culture tools. As I mentioned, we have lab robotics. We have internally generated data capture modalities, technology stack, which is basically our supercomputer and our artificial intelligence. And then all of our experimental designs and the execution and analysis of that data is specifically designed. It's purpose-built data collection so that we're actually able to make meaningful decisions based off of the data. So how do we use all this data? Actually, it's very simple when it comes down to it. We take a lot of pictures. As you can see here, these are just some examples of some images. We do a lot of cell culture and all those experiments, and we take pictures. And as you can see, these are different cells, probably different treatments, different experiments, and visually you can see the differences here. But I think the unique thing, the way that we capture the data, is there are so many data points within these pictures that we can't visually see that we're actually able to use and utilize when we input it into our supercomputer and make decisions based off of that data. And we're evolving and we move to the point where we can actually now, based off of the data set that we currently have, not necessarily pull data out of just these specific experiments, but we can actually ask questions to our supercomputer across the data sets to pull out specific information based off of whatever questions we're trying to ask related to a specific indication, specific disease, or specific set of compounds. And really what this is doing, and this is just a graphical representation, really our ultimate goal is to reduce the amount of time that it takes for us to go from, screening a whole bunch of compounds or drug candidates and getting to the point where now we're evaluating and assessing and testing a single compound to put forth into clinical development. And really all this picture shows is kind of the classic funnel design that the typical pharmaceutical companies go through, here in the red going down into the orange. And really what we're trying to do is kind of expand the opening of the funnel and shorten the time that it takes us to get down to a single drug candidate and moving that forward into development. And we've actually already shown that we can dramatically reduce the time that it takes for us to get to a single drug candidate in relation to what typically is done in most pharmaceutical companies. In this slide, this is just a snapshot of the programs that have resulted from our discovery approach. They range across a wide variety of diseases, including everything from obviously rare genetic disease disorders like GM2 to more common diseases such as cancer, as well as several other areas of medicine. And if you look over onto the right, you can see GM2, where we have listed among our currently active clinical programs. So that's just some basic information about our company and how our discovery approach works at Recursion. I know it's a lot to take in. It definitely is a new way and a new thought process of how to, to find and develop drugs for these indications. But what I would like to do now is talk to you a little bit about how we identified this particular drug candidate as a potentially promising fit for GM2. So I know that for this audience, I don't really have to explain the particulars of GM2. Really, if anything, the NTSAD and the patients and the doctors have really been a great partner in helping educate us about what life is like with GM2. I put this slide up just because I want to touch briefly on some of the basic biology of GM2, just to refresh everybody as we walk through Recursion's findings related to this. As you know, mutations in Hex A and Hex B lead to an enzyme deficiency that causes a buildup of some of the materials in the cells, and ultimately that buildup ends up being toxic to these cells, specifically the cells in the central nervous system, the brain, and the spinal cord. And this results in the clinical signs and symptoms that we see in GM2 patients. So this slide, this is some of our data that we've generated. So this is after we have identified REC 3599 as our drug candidate for GM2. And really what I want to do here is show how this drug is affecting. These are, we've actually taken patient cells. So these are actual infantile patient cells from a Sandhoff patient and a Tay-Sachs patient here. And what you can see on the far left, this is a normal cell. It's stained so that you can see the nucleus of the cells. So those blue circles are the nuclei. And it indicates that those are all live, viable cells. So everywhere you see those blue circles is the nucleus, and that's where the genetic material for these cells is located. And then moving to the middle and to the right for each patient cell line for the Sandhoff and Tay-Sachs, what you see in the left pictures, where you see the yellow state, we have a special stain specific for GM2. And you can see that these cells, without treatment, you actually see a pretty significant amount of GM2 present in the cytoplasm of those cells. And what we've shown is that when we treat these cells with our drug candidate REC 3599, we're able to significantly reduce the accumulation of GM2 in those cells. And we've also done some additional experimentation, as shown on the graph just below, where we actually do what we call a dose response, where we increase the dose of the drug, ultimately trying to get to a point where those GM2 levels are approaching the levels that we would normally see in healthy cells, that's indicated down in the line in blue. So really what I would like to do now for the last few minutes here is to kind of give you a preview of our upcoming GM2 clinical trial. As I indicated, REC 3599 is our lead drug candidate. So interestingly, this was actually a drug called riboxystorin that was previously developed by Eli Lilly. It was developed for complications to diabetes, specifically diabetic retinopathy. And they actually got pretty far in their development. And this drug candidate has actually been tested in adult patients, up to 2,500 adult patients. So we actually have a pretty robust clinical safety data set for this compound. Obviously, safety in patients is one of our primary endpoints for our programs. So this was really great for us. And as I've shown, we have potent activity in those cells from patients from both Tay-Sachs and Sandhoff, and we've done that assay with many different patient cell lines. So not just those two. So we've actually confirmed that this works in many patient cell lines. We're currently developing a liquid medication administered through a feeding tube. This will be a once daily administration. Our clinical development plan for REC 3599, we've talked with many people, including folks here at the NTSAD, as well as a lot of key doctors and patients, as well as many interactions with the FDA. We have been granted rare pediatric disease status by the FDA, and we're currently working very hard to potentially launch an open label study in GM2 patients starting within the next year. So at this point, I guess I'll like to thank everybody, thank the NTSAD for the opportunity, and I'd be happy to take a few questions in the time we have remaining.
So Joel, thank you very much, and very exciting use of technology there. Um, so a lot of talk about substrate reduction and the biomarkers there, but when it comes right down to it, the families want to know, are these going to help with symptoms? And obviously you're not in the clinic yet, but what can you say about any sort of expectations about preclinical or whatever your designs are telling you about how the biomarkers could translate into symptom improvement?
Yeah, no, that's a great question, and I can't say that the functional endpoints in a clinical trial are obviously going to be critical efficacy endpoints, and those will definitely be considered in our clinical trial. This will not just be a GM2 biomarker trial. Obviously our clinical designs will be announced a little bit further down the road, but that's definitely a primary consideration for us as we're designing our trial.
Thank you. And then one other question, it's more of a broader question, and I think we'll use this to transition to our next speaker. For those of the families out there that are hearing so many different types of therapeutic options, the good news is we didn't have a lot of options in the past. The challenge is trying to figure out which makes sense for you as you look at these things. So I think that's a big part, as we talked earlier about the role NTSAD is going to play in terms of sort of helping go through this clinical journey together, provide you, access to information, work with your healthcare providers to make sure you're getting all the information you need to look at what are the different options that make the most amount of sense to you. So I think at this point, it's a fundamental question, and the organization's here to support you. And we're going to move on now to Thank you very much, Joel, to Taste Gene Therapies, and they're going to